everybody, welcome to another video about Steam. Wait a second, I don't think you can see me. Okay. Is this? Okay, that's the correct button. Okay, there we go. Okay, hi everybody, welcome to another video about Steam. Stop, Amy! Okay, anyways, first as a quick shout out to today's...
You know what?
Stop! Okay. Anyways, as a quick shout out, today is my sister's birthday, so... Stop! So today's my sister's birthday, and please wish her a happy birthday down in the comments below. So the first thing I might notice is that there's this bowl on my table with a pair of chopsticks, and if you look inside, there's actually a lump of dough in there, and the reason I have this is an example of my failed experiment trying to make a cell similar to last time. Well, I don't know what happened, but it just didn't work. Anyways, so in order to create today's experiment, what I did was I just, took borrowed a clip from last video, and here it is. Itself, it needs to be torn apart like this. Itself, it needs to be torn apart like this. Okay, so now into talking about what we're doing today. So today we're talking about meiosis. Now you might be thinking, wait a second, didn't we already talk about mitosis? And then I was like, yes, but meiosis is not mitosis. Yeah, the two are incredibly similar, but there's one crucial difference. Well, there's multiple differences, but whatever. So, mitosis creates two cells. So you have like this original mother cell, and it creates two daughter cells. And those daughter cells are completely genetically identical. Now, genetics is something we'll be talking about later. And the meiosis, however, meiosis creates four non-identical cells. I almost said identical, that was incorrect. But four non-identical cells, and in fact, they're not body cells anymore. Because mitosis produces body cells. Like meiosis creates how should I phrase it to make it not inappropriate? Well, sperm and egg cells. Anyways, so that is basically what meiosis is, except we'll go into a bit of detail later. And well, by later, I mean right now. Alright, so here we have two cells, and there is one difference between the two cells. Can you spot it? You spotted it, it's because this one has a 64 in the nucleus, or this thing, and this one has a 32. Also, this one says diploid, and this one says haploid. We'll get to what those mean later. So, can't pronounce it. Is it diploid or diploid? I think it's diploid. Anyways, diploid cells basically have 64 chromosomes, well, humans. Humans have 64 chromosomes, by the way. Remember that. So a diploid cell basically has 64 chromosomes here. So in the nucleus it has 64. A haploid cell, however, only has half. So for example, in an organism, in a theoretical organism that only has 2 chromosomes in its diploid form, or diploid form, I don't know how to pronounce it, in its haploid form it will only have 1 chromosome. And also you might note that these two are exactly the same. Whoopsies. Wrong thing. These two are exactly the same. In fact, I copied over this one and then changed the number and changed the die into a half. So anyways, so now that we've understood what diploid and haploid are, let's go over here. So, we're going to be talking about meiosis 1 first. So, you have your ordinary cell, and similar to mitosis, it has gone through interphase and is ready to undergo meiosis, which is different from mitosis. And I'm going to draw the nucleus here. I'm going to draw the nucleus extra big because there's some stuff happening in the nucleus that is a bit special, and I don't think the nucleus is actually this big in real life, but I'm just exaggerating the size. Anyways, now that I've got this, and then, so let's say that we have chromosome here, their characteristic X shape, and another chromosome is here. And with this, something is going to happen during, anyways, so then in the first part of meiosis, which is called prophase, and it's actually very similar to the mitosis prophase, except one thing is that they can undergo cross-linking, or, wait, what is it again? I forgot. Anyways, cross something, but you should search up the phases of mitosis to get more depth into this. This is only the basics. Anyways, so this these two are just gonna go hanging out until they actually cross. So I'm gonna redraw this. I'm gonna delete that. I'm also gonna delete this because I don't know why I left that around. Anyways, so then it's almost like they're shaking hands. One is here, and they're like this. They look like this now. And let's zoom in on them. And say that this one is blue actually, so I'm gonna redraw them down here. And then this one is red. So we see them, and then they're crossing or whatever. I'm gonna delete this. And I'm gonna move this one up here. I know this is gonna leave some red behind, so I'm gonna erase that. I'm sorry I have to do this. Anyways, so then we have this. And then something really weird happens. So, remember that, the blue chromosome is done like this, and it actually has a bit of red on it. So, whoopsies. Forgot to select red. And so now it looks like this. And now the red chromosome has a bit of blue in it. Like this. And this is so that the resulting sperm or egg cell will have more diversity. Okay, anyways, on to the next phase. Actually, I should probably draw the centrioles here. You have one pair of centrioles here, one pair of centrioles here. They're all, these stuff are going over here. Spindle fibers again. Okay. On to metaphase. Metaphase. This is our cell in metaphase. And I should zoom out a bit. There. So, and again, this next part is really similar, so I'm just gonna go ahead and review it with you. It's gonna have two of the centrioles are lined up here, and then there's one thing that's different. Instead of these chromosomes lining up on the middle like this, instead of doing this, they do this. So they actually line up along the same axis as I should redraw that, but they actually line on the same axis as the centrioles. So they look more like this. And yeah, I should better draw them like that because that's actually how they are formed. But not like this, but more like this. Anyways, and of course there are spindle fibers here, and there's spindle fibers here. And that didn't do a very good job of connecting. But anyways, and then we have anaphase, which is the away phase. And yeah. Anyways. And then this one I'm intentionally going to draw a bit fatter because after doing my last video, I found out that anaphase is actually the moment where the cell starts to be squished in. And yeah, so then we have anaphase, we have the centriole here, and then we have this thing, and the chromosomes are being pulled apart. Bye-bye. And yeah. Also, by the way, this is done in a theoretical 2-chromosomed cell, which doesn't exist, obviously. And anyways, finally telophase. Now, this is actually, I've been saying this incorrectly, it's not actually just prophase, anaphase, prophase, metaphase, anaphase, telophase. It's actually, these are prophase 1, prophase 2, prophase 1, metaphase 1, anaphase 1, and telophase 1. And you'll see why later. So then, the chromosome is here, and the chromosome is here. Now one thing to note that this thing hasn't replicated its DNA. And it used to be this is actually its diploid form. And the resulting 2 cells, which are again called the daughter cells, they're actually haploid. So they actually just cut the amount of DNA they have inside of them in half. So it's basically like you took this dough ball. I don't think it's a ball, but whatever. This lump of dough, and you split it in half without allowing it to grow, like I did in the last time. Actually, I didn't grow it. I just added more. Flour than I did this time. Anyways, of course you have your centrioles here, and this one has no room. Oh no. And I should probably draw that a bit better. It looks more like this now. And yeah, it's time for cytokinesis. And cytokinesis is basically what just happened. They form into 2 daughter cells. And these are not identical, okay? Because what happened in anaphase and metaphase makes it different from in regular metaphase and anaphase. Well, should I say regular? I don't think this is, well, I don't think this is regular and not regular. Anyways, I'm going to move back here, and then I'm going to do a side-by-side comparison of mitosis and meiosis in metaphase and anaphase. So, and so then we have these, and in metaphase mitosis, this is how they line up. And in anaphase mitosis, this is how they start to pull apart. And I'm going to also draw it a bit oblong, like this. And there we go. I'm going to use the brush tool again. I'm sorry I'm taking too much time here. I might delete this drawing part because it's too long. They do this. And so there's a massive difference here. And as you can see, this the metaphase and anaphase and mitosis are where they line up perpendicular to the axis of the centrioles. So in case you don't know what perpendicular means, it's basically 90 degrees to each other. And in meiosis, however, they just line up on the same axis. So like this. Anyways, back to cytokinesis. And now that we have our 2 cells, like this. And yeah, those are our 2 cells. We also have the nucleuses. That's obvious, of course. We have the nucleuses. I'm not going to draw the DNA anymore. But anyways, and then you might remember me saying that it created 4 cells, and you might be saying, wait a second, this is only 2 cells. And yes, that is right, because now we move on from meiosis 1 and go into meiosis 2. I keep saying mitosis when I try to say meiosis. Anyways, and meiosis 2 is very similar to meiosis 1, except none of this stuff. None of like yours is now mine and mine is now yours. That kind of stuff. None of that. It's just plain old meiosis without this. Anyways, well, that's about it. Alright, it's time for the question of the video. So what happened if there's a mutation in this? Yeah, mutation. Mutation is when the genetic code is incorrect. And it prevents, it duplicates its DNA accidentally. What is going to happen? Is it going to stay with 64? Is it going to stay diploid all the way down, or is it going to somehow turn into a haploid? Leave your answers in the comments below. Please like and subscribe, and I'll see you in the next video.